Hi, my name is Mitchell Harrison, and today I'm going to be talking to you guys about monoclonal antibodies. So, developed in 1998, trastuzumab is a humanized monoclonal antibody that significantly improves overall survival of HER2-positive early breast cancer. The median time to see patients after their treatment is 11 years in a follow-up. So in a large study with 5,102 HER2-positive early breast cancer patients, Herceptin was applied to either patients after primary treatments of chemo, surgery, or radiotherapy for 1 year, 2 years, or not at all to determine the effect it had, and they were testing to see the endpoint of progression-free survival. Here is a figure from the research showing the incidences of disease-free survival depicting the number of cases showing positive effects for the treatment on the right-hand side, and we can see the percentages increase with the amount of time that trastuzumab has been applied to these patients. So breaking it down, we can see the 1 year treatment significantly improved the 10-year survival rate of patients just as much as the 2 year treatment. So there's no additional treatment benefit for 2 years versus 1 year. But it should also be noted that 52% of the patients in the observation group switched over to receive the Herceptin treatment. It doesn't clarify as to why they switched, but it may be because their cancer was not healing and they wanted to see better results. So in conclusion, we could say that the 1 year treatment of trastuzumab after chemotherapy for patients with HER2 positive early breast cancer significantly improves disease-free long-term survival, and the 2 year treatment will have no additional benefit for extended treatment. Next up, we have rituximab and hyaluronidase human in a subcutaneous formulation of the rituximab that has been humanized. The intravenous is chimeric, and this targets the CD20 transmembrane protein. It has been approved to treat follicular lymphoma, diffuse large B-cell lymphoma, and chronic lymphocytic leukemia. But today we're just going to be focusing on the follicular lymphoma. This drug attacks cancer cells through direct drug-mediated signaling, complement-dependent cytotoxicity, and antibody-dependent cellular cytotoxicity. So that means that it could either prevent these proteins from accessing or preventing T cells from coming in. It could also poison the cell and kill it, or it can also trigger apoptosis as well. So the name for the study that we're looking at is going to be the Sabrina trial, and it was a randomized phase 3 study comparing rituximab intravenous versus rituximab subcutaneous and hyaluronidase human in combination with standard chemo in 410 patients. In order to receive the subcutaneous treatment, the patients must had to first go through 1 round of the intravenous drug, so that way they have a base rate, and then they were testing to see if it had any further effect in terms of efficiency as well as cost efficiency. They tested to make sure the subcutaneous method did not show inferiority to the intravenous version, and they tested the response rate, which showed that 84.4 to 84.9% of patients saw a positive effect from their treatment. There were also side effects that consisted of neutropenia, which is a low white blood cell count in 27 to 32% of patients with the IV and the subcutaneous version respectively, and administration reactions in 48 to 35% of patients respectively as well, usually just irritation and itchy skin. So our next monoclonal antibody is going to be durvalumab. It's a PDL1 protein so it binds to PD-1 receptors on cancer cells. And what that does on T cells, the PD-1 receptor is like a brake on the T cell, and this protein switches that off. This has been approved by the FDA to treat non-small cell lung cancer, and that's what the study we're looking at today is going to be looking at. All of these patients in this study had stage 3 NSCLC and were split up into either the test group or the placebo group at a rate of 2 to 1 respectively. All of these patients had not seen growth after having undergone 2 or more cycles of platinum-based chemo, and they were testing the progression-free survival and overall survival as their main endpoints in this study. So as you can see here, this is a picture of a graph from the study showing the tracking the probability of progression-free survival over time since the randomization, and you can see that the blue line of the durvalumab versus the placebo is much higher, leading us to believe that our results will show us that durvalumab will have a significant increase in this probability of progression-free survival. So following the graph from the last slide, as we can see from the results, they're pretty staggering in terms of empirical data showing the drastic increase in progression-free survival. Um, Same with the response rates to the medication and the increased average time to death, with the durvalumab and the progression-free survival being 16.8 months versus 5.6 months. That's the amount of time that they have the cancer inside of them, but it doesn't grow or change. It's just there. So things tend to hold more steady with the durvalumab. The progression rate is also greatly increased from 16% with the placebo to 28.4% with this drug. And also, this increases the time of death by just under double the amount of time from the placebo. So, as we can see, the empirical data here really shows that durvalumab is very beneficial to treating this type of cancer. There also were some side effects like pneumonia in 4.4% and 3.8% for durvalumab and placebo, respectively. But besides the adverse side effect, the results are quite promising. So our last receptor that we're going to look at is the dinutuximab, and it's a chimeric, and it attacks the GD2 receptor on cancer cells. This receptor is found in human cells, but its expression is restricted to the brain primarily. The total incidence of GD2 positive tumors is greater than 200,000 annually, and these cancers have a high mortality rate between 20 to 80%, where 60 to 100% of them, based on the tumor types, are candidates for this anti-GD2 immunotherapy. But the cancer that we're going to be looking at today with this particular signaling is going to be the neuroblastoma. So this study looks at a treatment of 35 patients with neuroblastoma, 18 of which followed the control group for the treatment, while the other 17 followed the same treatment but with the addition of dinutuximab. The primary endpoint was the proportion of patients achieving an objective, complete, or partial response by a central review committee after 6 cycles of this treatment. As we can see from this table of the control group, one of the patients achieved a partial response. This is compared to our dinutuximab group, where of the 17 tested, 9 of the patients received or achieved objective responses, with 4 partial and 5 complete responses. The side effects include the neutropenia in 44% of patients, or 8 of the 18 patients, and anemia in 6 of the 18 patients in that control group. And the side effects for the dinutuximab group included pain for 44%, or 7 out of the 16, hypokalemia, which is found in 6 of the 17, fever and infection in 4 of them, and neutropenia in 4 of them as well. The results show that there is promise in this drug, but it does merit more research since the sample size for the groups were significant or statistically insignificant. But just from the initial testing here, we see a lot of promise going into this drug. Looking back, we see the results shown in each of the studies provided for the monoclonal antibodies. We can see how they interact with different receptors of cancer cells and how they may not all function in the same regard to targeting their specific cancer cells, but some of them block receptors, others add in cytotoxicity. But from these interactions, we can see that the results are quite promising for these particular drugs. Even in cases that examine stage 3 and later developed cancers, we see an increase in reaction and remission. These findings show that greater research is required into these monoclonal antibodies to be favorable to further develop them and make them more effective in fighting these cancer strains. But that being said, this also includes more research into the side effects of these treatments and how they combat things like pneumonia that could pop up in certain cases, and also like low white blood cell counts and how we can fix that while still fighting the cancer. But overall, the studies have proven promising, and we have great potential looking forward with these particular drugs. And here at the end is just a list of references that I use to go through and find my research for this PowerPoint.